Have you ever wondered how doctors can identify where a cancer began, even after it has spread throughout the body? A tumor discovered in the liver doesn't always mean liver cancer, and a mass in the lung isn't necessarily a primary lung tumor. One of the most important jobs of a pathologist is to answer a deceptively simple question. Where did this cancer come from? The science that answers this question is called histogenesis, and it lies at the heart of modern precision oncology. Histogenesis refers to identifying the original tissue or cell type from which a tumor developed. This information is essential because cancers arising from different organs often require completely different treatments, even if they appear in the same location. Correctly identifying the tumor's origin leads to a more accurate diagnosis, a better prediction of prognosis, and ultimately the most appropriate therapy. This becomes especially important when cancer metastasizes. Metastasis is the spread of cancer cells from their original location to distant organs through the blood or lymphatic system. A patient may present with tumors in the liver, lungs, bones, or brain, while the primary tumor remains small or even undetected. In these situations, simply knowing where the tumor is found is not enough. The real challenge is determining where it started. To solve this puzzle, pathologists combine several powerful techniques. The first is traditional histopathology. Under the microscope, pathologists carefully examine the tissue architecture, how cells are arranged, how they interact with one another, and whether they resemble normal tissue. Different organs have characteristic microscopic patterns, and experienced pathologists can often recognize subtle clues that suggest the tissue of origin. However, many cancers lose their normal appearance as they become more aggressive. In poorly differentiated tumors or metastatic cancers, the cells may no longer resemble the tissue from which they arose. This is where one of the most important advances in modern pathology comes into play: immunohistochemistry, commonly known as IHC. Immunohistochemistry is a laboratory technique that uses specially designed antibodies to detect specific proteins inside cells. To understand how it works, think of antibodies as highly specialized molecular detectives. Each antibody is designed to recognize and bind to one particular protein, much like a key fitting only one lock. Once the antibody binds to its target, a chemical reaction produces a colored stain that becomes visible under the microscope. The pathologist can then determine exactly which proteins are present inside the tumor cells. These proteins serve as molecular fingerprints of different tissues. Instead of relying only on how the cells look, pathologists can identify what the cells are producing. This dramatically improves diagnostic accuracy, especially when tumors have spread to distant organs or have become so abnormal that they no longer resemble their tissue of origin. Even more importantly, pathologists rarely rely on a single marker. Instead, they use a carefully selected panel of immunohistochemical stains. Just as detectives combine multiple pieces of evidence to solve a case, pathologists interpret patterns of positive and negative staining to narrow down the tumor's identity. One marker alone may suggest several possibilities, but a combination of markers often leads to a highly confident diagnosis. Today, immunohistochemistry does much more than identify where a cancer originated. Many IHC stains also reveal proteins that predict how a tumor will behave or whether it is likely to respond to specific targeted therapies. In other words, IHC helps answer not only what type of cancer is this, but also which treatment is most likely to work. This is one of the reasons immunohistochemistry has become one of the cornerstones of modern precision oncology. Increasingly, molecular profiling provides another powerful layer of information. DNA sequencing and gene expression analysis can identify mutations or molecular signatures that are characteristic of particular tumor types. Together, traditional histopathology, immunohistochemistry, and molecular diagnostics provide one of the most accurate diagnostic systems in modern medicine, allowing physicians to identify a tumor's origin with remarkable precision. The clinical importance of histogenesis becomes obvious when we look at real examples. Imagine a patient with a tumor in the liver. At first glance, it may seem like primary liver cancer. However, the liver is also one of the most common destinations for metastatic tumors, particularly those arising from the colon. Under the microscope, together with immunohistochemical markers, pathologists can distinguish hepatocellular carcinoma from metastatic colorectal adenocarcinoma. This distinction completely changes the diagnosis, prognosis, and treatment plan. A similar challenge occurs in the lungs. Not every tumor in the lung is the same disease. Most lung cancers are epithelial carcinomas that arise from the cells lining the airways. In contrast, mesothelioma originates from mesothelial cells lining the pleura surrounding the lungs and is frequently associated with asbestos exposure. Although both diseases may involve the chest, they arise from different cell types, behave differently, and require completely different therapeutic approaches. To identify these tumors, pathologists rely on tissue-specific biomarkers. One of the most commonly used groups of markers is cytokratins. Cytokratins are structural proteins found inside epithelial cells, the cells that line the skin and most internal organs. Because nearly 80 to 90% of all human cancers arise from epithelial tissues, most carcinomas stain positively for cytokratins, immediately indicating an epithelial origin. Another important marker is CD20. CD20 is a protein found almost exclusively on the surface of mature B lymphocytes. When a tumor stains strongly for CD20, it indicates that the cancer originated from B cells, helping diagnose B-cell lymphomas such as diffuse large B-cell lymphoma. Melanomas require a different set of markers. Proteins such as S100 and HMB45 are expressed by melanocytes, the pigment-producing cells of the skin. Their presence strongly supports the diagnosis of melanoma rather than another type of cancer. TTF1, or thyroid transcription factor 1, is another highly valuable marker. Despite its name, it is normally expressed in both lung and thyroid epithelial cells. When a metastatic tumor stains positive for TTF1, it strongly suggests that the primary tumor originated in the lung or thyroid. Many additional tissue-specific markers further refine the diagnosis. PSA is highly specific for prostate tissue and helps identify metastatic prostate cancer. PAX8 is commonly expressed in tumors arising from the kidney, thyroid, ovaries, and other Mullerian organs. GATA3 supports the diagnosis of breast and urothelial cancers. HepPAR1 helps identify hepatocellular carcinoma, while Chromogranin A is a classic marker for neuroendocrine tumors. Each marker contributes another piece of the diagnostic puzzle, and together they allow pathologists to identify a tumor's origin with remarkable precision. Correctly identifying the cellular origin has enormous clinical consequences. Once the primary tumor is known, physicians can select the most effective targeted therapies, estimate how aggressively the cancer is likely to behave, predict prognosis more accurately, and design an individualized treatment plan. Without knowing where a cancer began, choosing the right treatment becomes far more difficult. This is why histogenesis represents one of the foundations of precision medicine. Behind every biopsy lies an investigation that combines microscopic examination, immunohistochemistry, and molecular biology to answer one of oncology's most important questions. Not simply, is this cancer? But, where did this cancer begin? Because only when we know the tumor's true origin can we offer the right diagnosis, the right treatment, and ultimately the best possible outcome for the patient.